Start broadcast.
The broadcast is now starting. All attendees are in listen-only mode.
Good afternoon, everyone, and thank you for joining us this afternoon for the latest installment of the HDSA Research Webinar Series. This is George Orling from the HDSA offices in New York City, and I'm happy to have a fantastic speaker lined up for you for this afternoon, Dr. Francesca Cicchetti from the University of Laval. But before I introduce Francesca, just want to go over some of the instructions. Sorry about that. About how you can ask questions if you are new to these research webinar series. In the right-hand side menu box where it says questions, you can just type any question you may have for either myself or Dr. Cicchetti at any point during her presentation. And when she's done, we'll ask all of them at the end. Question and press send. Alternatively, if you have to dial out, or there are other people you think may be interested in hearing this presentation, this is being recorded and will be archived on our website. Within a week or so, you'll be able to find it on hdsa.org, where it'll direct you to our new YouTube channel, where you'll be able to see this webinar. Just a real quick update on our upcoming schedule. Next month, I'll actually be presenting a demonstration of our new clinical trial matching tool or finder called HD Trial Finder. It's an online matching service for the HD community. It was just released last week in conjunction with our new hdsa.org website, and I'll be giving a demonstration as to how to use this new tool. And then in June, and we will not be having a webinar in June in lieu of the HDSA Convention Research Forum, which will be taking place on June 27th from 10 to 1 p.m. Eastern, and we'll be webcasting that for the entire community. So if you can't join us in Dallas, you'll be able to participate in home mornings full of science in June. So let me just want to introduce Dr. Cicchetti. Dr. Cicchetti is a currently professor in the Faculty of Medicine at the Department of Psychiatry and Neurosciences at the University of Laval in Quebec, Canada. Before that, she was a visiting associate professor in the Department of Radiology at Harvard Medical School. She received her PhD from the University of Laval and also studied and did her postdoctoral fellowship at Harvard Medical School. Her work focuses on the development of therapeutic approaches for neurodegenerative diseases, and in particular, she'll be talking to you about how huntingtin proteins spreads from cell to cell and the implications that this has on potential stem cell transplant therapies for Huntington's disease. So without further ado, I'm going to turn it over to you, Francesca.
Okay, so am I on, George?
You are on, and we see your screen.
Okay, perfect. Thank you. Thank you, everyone, for attending this webinar. I'm Francesca. Before I begin, I do want to thank George and the Huntington's Disease Society of America for giving me this unique opportunity to present some of the work that we've done in the last decade and even the last 15 years in my lab. I also want to congratulate Huntington's Disease Society of America for initiating these webinars. I think I speak on behalf of many of my colleagues, fundamental researchers, when I say that for us this is truly a unique opportunity to talk to you, but an invaluable dialogue to hear your comments, your feedback, your questions, which I think will really help us push the work forward and hopefully translate these results into meaningful therapies in the near future. So I'm very grateful for that. I'm just having some difficulties. Okay. As the title of my talk implies, I will be discussing today the data that we've collected using postmortem analysis of the brains of Huntington patients who received cell replacement therapy in the hope to stop the progression of their disease. My main, and I'm going to be summarizing actually 15 years of data, so I'm going to try to do this in 30 to 40 minutes. But I have two main goals by doing this. If you want to switch the slide, George, I don't seem to be able to do this.
Just click on the right arrow down, and yeah, you should be able to do that.
Okay, thank you. Thank you. The first objective of this webinar is really going to be to review all the observations that we've collected and how this and what this means for cell replacement therapy in Huntington's disease. And I'm going to give you my honest opinion on what we should do with this experimental therapy, whether we should take it forward, and I will be happy to hear your views on that as well. And the second point, or the second objective, will be to take these observations and really show you how much they've helped us understand the pathology itself. Every data that we've collected has been tremendously insightful into the pathology of Huntington's disease, and this is really the point that I want to review with you. So as you all know, Huntington is driven by a single gene, and this gene will lead to the mutation of the huntingtin protein, which is a protein that's very important in development but also in normal brain function. When the mutation occurs, you start to see these aggregates of the mutant huntingtin protein, which are found in the cytosol and the nucleus of the cells. As you can see here, these black dots are all inclusions that are found in the brains of a patient with Huntington's disease. Now, as I mentioned, these mutations are found in every cell of the body. They're found in the muscle cells, they're found in the endocrine system, they're found in cells associated with the blood, they're found in fibroblasts. So this mutation, of course, will alter the function of the cell that it's found within. And because of that, we know that Huntington's disease is a fairly complex disorder. It doesn't just involve the brain, but it involves other peripheral problems such as muscle atrophy, cardiac failure, and so on. So it is a complex disorder, but the research over the years has mostly focused on the brain because we know that the impairments in the brain lead to the cognitive problems, the psychiatric problems, but also the motor impairments that are the main symptoms. Now, from a neuropathological perspective, you can see that in the brains of these patients, when you study it, there's a major atrophy of many of the structures of the brain, and more particularly the structures of the basal ganglia, which are responsible in large part for movements, but also some aspects of cognition. So you can see that with advancing HD, you have this atrophy of the structure that's mainly due to the degeneration of neurons that project outside of the structure, the striatal projection neurons. But the interneurons, the neurons that are involved in the local communication, are actually mostly spared. So thus far, a lot of experimental therapies, of course, have been aimed at targeting these brain abnormalities and these brain impairments. So far, there's been attempts to deliver growth factors, for example, or try to promote adult neurogenesis. But at the forefront of these therapies has been cell replacement therapies or cell transplantation, and I'm sure you've all heard of this. I mean, it's been extensively talked about, especially with the discovery of stem cell and iPS cells. So the idea of cell replacement therapy is that, as I mentioned to you before, you have primarily, not exclusively, but you have primarily a loss of striatal neurons, which of course will lead to the degeneration of these connections to other structures of the brain. So the idea of transplantation is to replace the lost cells by putting new cells into the striatum, hoping that not only will they start to release the neurotransmitters that are needed for the function of the structure, but also start to form connections to other structures of the brain, which will reestablish normality in terms of movement and cognition. So far, there has been 6 or 7 independent pilot studies. It depends how you view them, but let's say there has been 7 independent pilot studies that have addressed the efficacy of cell replacement therapy for Huntington's disease. You can see most of these studies took place over a decade ago, and you can see that most of these studies have recruited very few number of patients. They're all pilot studies. And you can also see that the methodology that was used across these trials is very different in terms of the tissue that was used, the method that was used, also the immunosuppression regimen that the people were put on. So it becomes a bit difficult to compare the outcome of all these trials, but I think it's very important that we make the most of the data that has been published, and you can see and you can look up these papers. There's been a number of reports on the surgical procedures and the clinical data of these trials, on the clinical outcome and the imaging studies derived from these trials, but also on the post-mortem studies. I mean, you can see that we've been involved quite extensively in this, but other groups have also published their data on this. So to some extent, we're able to compare the data across these pilot studies and see what benefits these, the cell replacement therapy has been for Huntington's disease. So today I'm going to talk specifically about this trial. This is the one I've been involved with. It's a trial that took place approximately 15 years ago at the University of South Florida. It was carried out by Thomas Freeman, a neurosurgeon. And you can see that this was the largest clinical trial that took place. It recruited 7 patients in total. Now, out of these 7 patients, 5 of them have come to autopsy. You can see on this table that one patient passed away 18 months after the surgery of causes unrelated to the procedure, but most of the patients passed away many years after the transplant. So this, of course, gave us an opportunity to compare the behavior of the transplanted cells early after the transplantation, but also very late after this procedure, and see how these cells behave long-term in the brain of someone that's affected with Huntington's disease. So before I get into the core of the data, I do want to explain to you the methodology of transplant if you're not familiar with it. The idea is to take embryos at an age of about 8 to 9 weeks post-conception, and we dissect an area of the brain that is known to give lots of striatal cells, so the cells that we're trying to replace. This is the area that we dissect. Now, when we do this dissection, there are 2 ways to transplant the cells. You can mechanically dissociate the cells and transplant them as individual cells into the brain, or you can keep them as whole pieces of tissue, and the idea behind this is that we want to keep the microenvironment of the dissection intact and not perturb the cells that we've dissected, and then we transplant these pieces of tissue into the brain. Now, in the trial that I was involved with and the analysis that I'm going to be showing today, it's all whole pieces of tissue that were transplanted. So this here is exactly an example of this transplant. This is the putamen, so the structure where we transplanted this tissue in a Huntington's disease brain. And you can see right here, I'm going to show you with this dotted line, this is the transplant. So it's very easy to identify because it's a whole piece of tissue, the anatomical borders are very easily identifiable, and this is what you see here. Now, every piece of tissue here that's darker, it's darker because it's stained for striatal markers. So all of these areas express cells that have the phenotype of striatal cells, the one that we're trying to replace. All of the in-between pieces are tissue that are expressing microglial cells or astrocytes. So you can see that within this dissection, most of the tissue, the great majority of it, is actually striatal tissue, so the tissue that we're trying to replace. So here, if you compare the brain of a patient at 18 months post-transplantation and a patient at 10 years, you can see that all of these grafts have survived and that the tissue within it is expressing different markers of striatal tissue. And you can see that this is also true at 10 years. So the tissue seems to be surviving long term. But when you look more closely at these cells, the transplanted cells individually, what you can see at 18 months is that these cells are very intensely stained for markers of striatal cells, and you can follow their dendrites for a number of micrometers in a neuropil, so they look fairly healthy. But when you compare this to grafts, 6 years, 7 years, or 10 years after transplantation, and this is actually the work of another group in California, you can see that the cells are not as intensely stained, and they don't have these long dendrites and axons that allow them to make synaptic connections. Now, this is not an artifact, because if you look in the same, with the same staining in the cortex of the same individual, these cells are very healthy. So it's very specific to the grafted cells. So there seems to be a degeneration pattern. The transplanted cells are degenerating in the same way that we see the cell degenerating in the brain of a patient with Huntington's disease. And if we look more closely at these cells, you can see that they have a very different morphology than the cells in the brain. They're actually more rounded, they lack this structure in the cytoplasm, and they even stain positively for caspase-3, which is a marker of apoptosis, so cell death. And cell death is a prominent feature in Huntington's disease as well. Another thing that we looked at was connectivity. Of course, when you transplant the cells, you hope that they're going to start making connections and crosstalk with the host, with the brain host. And we looked at that, and other groups have looked at that, and we were able to see that the graft, which is here, is receiving connections from different structures of the brain. And the synaptic activity within the grafted tissue is very high. So there seems to be contact of other structures of the brain with the graft. We even showed that the cortex right here was also forming connections with the grafted cells. And this is actually a synapse making contact with the grafted neuron. Now, despite the fact that this is, of course, good news, we see that the grafted cells can make contact with the host and vice versa, and that the two cell populations can talk to each other. We think that this is actually probably not so good for the grafted cells, because the cortical projections in Huntington's disease release an excessive amount of glutamate, probably causing what we call excitotoxicity and creating more degeneration of the grafted cells. So in this case, this may actually be detrimental to the grafted neurons. However, when we looked at interneurons, remember in the introduction I mentioned to you that interneurons were spared from degeneration in Huntington's disease, you can see that this is also true in the grafted tissue. So all types of interneurons are healthy, they have long dendrites, and they seem to be very healthy. So the neuronal sparing, the neurons that are spared, is actually very similar also to what we see in the brains of Huntington's disease patients. Another thing that we looked at was the inflammatory response. So microglia are a type of cells that are very important in releasing cytokines, so they can create inflammation and create degeneration, but they're also very good. Sometimes they can release neurotrophic factors and give support to, for example, grafted neurons. So here you have a picture of microglial cells which are in contact with grafted cells that I've pointed out with the red arrows. And what's interesting in this picture is that you can see that almost every grafted cell has a microglia attached to it. So there's a very close proximity of these two cell types. And sometimes the morphology of the microglia is very different. Here you have a microglia that has a morphology, a very activated cell, like a very angry cell. It's probably releasing cytokines and participating to degeneration. Whereas here you have a more resting microglia. Perhaps it's secreting neurotrophic factors and trying to save these grafted neurons. Another aspect that we looked at was the astrocyte. An astrocyte is a cell that's very important in also releasing neurotrophic factors and maintaining the health of the cell. And what we found is that there were very few astrocytes within the grafted tissue. You can see this is the work of other people. You can see within the graft you barely see any astrocytes. We saw exactly the same thing. But we saw some astrocytes just at the border of these grafts, cells that were mainly activated. So there is a reaction of the astrocytes around the graft, as if something, of course, is going on. They're probably reacting to degeneration. But it's not a glial scar. So, for example, fibers are able to get into the transplant and make connections with the transplant. Now, the fact that the astrocytes are not present in the graft, however, is important because astrocytes are very good to buffer glutamate. And as I mentioned before, we have this excessive glutamate release from the cortex. So if the astrocytes are not there to buffer it, there's a possibility that we're increasing degeneration of the grafted neurons. And again, astrocytes are important trophic support, and because they are not found in the graft, then we probably also have a lack of trophic support. Finally, we looked at vascularization. We were interested in seeing whether the graft received blood vessels and vascularization from the host in order to get the right nutrients to support its survival. And what we found is that there were very few blood vessels in the grafts. This is in the brain. You see lots of blood vessels, but very few in the transplant. We also saw that there was very little or very poor interaction between the astrocytes and the blood vessels, which create the blood-brain barrier. So the lack of integrity of the blood-brain barrier also exposes the graft to outside toxins. And this was verified by different markers which stain these tight junctions or the blood-brain barrier per se. So these are all very important observations for the grafts. In fact, I've summarized all of these observations in this schematic just so that we recapitulate all the findings that we have found so far. So I want to remind you that, again, the grafted tissue is receiving cortical projections, which is probably leading to an excess of glutamate. So these cortical projections are probably aberrant and creating more excitotoxicity. We've also shown you that there's a microglial response that is surrounding the graft, and this microglial response is possibly very toxic to this graft as well. We've shown that the astrocytes are not very present within the graft, so there's probably a lack of trophic support. We saw that there was poor vascularization and probably leading to less nutrients and less oxygens. And we also saw that there was poor communication or integration of the blood-brain barrier and the interaction between the graft and the host. So you can see here, and I don't need to emphasize this, but this data is, of course, challenging this methodology for Huntington's disease. You can see that there are a number of obstacles that we need to overcome in order for this transplant to be viable long-term, but also to yield benefits long-term. I did not address this, but we can talk about it at the end of the webinar. The clinical benefits seen in the patients that underwent this procedure are very mild, if not anecdotal, and they're short-lived. So obviously this is reflected by the fact that the grafts are not doing well in the brain over time. Now I'm going to come to more specifically the title of my talk, and that is the presence of mutant huntingtin protein in the grafts. So the story doesn't end there. I mean, we've already seen that there's a number of obstacles. We see that these grafts degenerate over time, but one of the questions that we were very, very interested in was whether the mutant protein could be found in the grafted tissue. And the reason for this is that because Huntington is purely genetic, it has been believed for decades that it's driven by cell autonomous mechanism. That means that the mutation creates the expression of the mutant protein within every single cell, so it's an intrinsic phenomena, and this is how the cell gets sick, an intrinsic phenomena. But there is more and more evidence in the literature, especially in the field of Parkinson's disease, in Alzheimer's disease, which are more sporadic disorders, that these proteins can actually travel from infected cells to healthy cells, as if they're behaving like a virus. And we were interested in seeing, even though this is a purely genetic disorder, whether the mutant huntingtin protein could also behave in this way. And this followed actually two very seminal papers that were published in Nature Medicine in 2008 by two independent groups, where they showed that the grafts, so the same type of procedure, grafted cells in the brains of patients with Parkinson's disease, if you looked at these grafts 10 years after the transplantation, the grafted cells also started to express the pathology just like you saw in the brain. So again, what about mutant huntingtin protein? Is it present in the grafted tissue? And if the theory that Huntington is governed by cell autonomous mechanism is true, then of course we should not find the mutant protein within the grafted tissue. However, we did find the mutant protein in the grafted tissue. This is the graft, again, very easy to delineate the anatomical border because this is a whole piece of tissue. You can see again those zones that are rich in striatal cells and the zones that are not so rich in striatal cells. And you see that within them you have these inclusions just like I showed you before. So these little aggregates are aggregates of mutant huntingtin protein that we found, and we found them everywhere in the transplant. Not just in the zones where there was striatal tissue, but also in the zones where there was no striatal tissue. These are the same inclusions in the cortex. You can see that they're not as numerous, and they may not be as big, but they're just as numerous and just as big as what we see in the striatum of the HD brain. So where did we find these inclusions? This is a staining. All the red cells are the grafted cells. And all these green dots are the mutant huntingtin protein. The blue staining here is a staining for the extracellular matrix. So we found most of these inclusions within the neuropil, in between the cells, if you like, of the grafted tissue. This was confirmed with a confocal microscope, where you can see exactly what these inclusions are embedded within the extracellular matrix. So they're not in the cells, they're outside the cells. This was confirmed again with different types of methods and antibodies, and we always saw these inclusions in the grafted tissue, as if they were being secreted by cells. Maybe when the cells degenerated, it left behind the protein aggregate. Now, in the brain of a Huntington's patient, the inclusions are found in the cell body, they're found in the neurite, and sometimes they're found to form this chain. You can see here several green dots, maybe it's easier to see it here, as if they're forming a pathway, as if they're following an axon or they're following a dendrite, as if they're traveling down this path. And we also saw the inclusions in the Huntington's brain, of course, in the extracellular milieu, as if they had been secreted by the cells. So I'm going to skip the next slides. It's not really important. This is just to show you, I mean, if you look again at the webinar, you can look at this in more details, but these are several techniques that we used to really prove that we could see the mutant huntingtin protein in the grafted tissue, because when we first wanted to publish this, people were very skeptical. And of course they were skeptical, and we needed to really prove this because it has huge implications not only for cell replacement therapy but also for understanding Huntington's disease itself. So I skipped the data. This is not so important. It was just a series of methods to show you that we did prove that the mutant huntingtin protein was found in the grafted tissue. Now I'm going back to the very complex picture that I showed you before, which summarized years of research looking and studying these grafts. And now we're adding to this already very complex picture the release of the mutant huntingtin protein in the grafted tissue. Now, of course, for us as researchers, this has huge, we understood that it had huge implication for understanding the pathology of Huntington's disease. So we were very interested in knowing how does the mutant huntingtin protein spread from the diseased host and the diseased cell to healthy cells. Because remember that the grafted tissue is genetically healthy. It doesn't carry the disease, and we transplant it into this brain that has Huntington, and after years it starts to express the pathology. So how does this occur? Before we published this data, there was only three studies that had shown in vitro, using different cell lines, that mutant huntingtin could be picked up from one cell to another. And there was a single paper, this last paper, that suggested a mechanism by which this occurred. It suggested that nanotubes, so little bridges between cells, could actually take place, sort of occur between cells, and transport the mutant protein from one cell to the other. This is what is summarized in this table. And you can see that in other disorders like Parkinson's and Alzheimer, if you look at the proteins that are very specific to these diseases, these mechanisms have also been identified, but they're much further ahead of us in this field, and they have identified a number of other mechanisms that could explain the propagation of these proteins. And this is going to be very important because we're going to use some of these theories and some of this data to apply to Huntington and try to develop therapies faster. So we have two theories that we are most interested in to pursue to explain the propagation of the mutant protein. The first one is the transsynaptic spread. So remember, I showed you that the graft receives cortical projection, and we showed this with the method of electron microscopy. I showed you the synapse between the cortex and the grafted cell, and I showed you that there were a lot of aggregates that could form these chains and these pathways as if they were traveling from one structure to the other. And one thing I did not mention that's also very important is that the part of the cortex that specifically projects to the graft or the striatum is very, very rich in mutant huntingtin protein, more than other areas of the brain. So with this video, I've illustrated how we think this occurs. So over time, of course, and in here you're going to have a zoom into the cortex, or the layer 5 of the cortex, you have the accumulation of the mutant huntingtin protein. So at first the mutant huntingtin protein is in a soluble form, and then it starts to shape up into these small aggregates, and then it forms these larger aggregates. And despite the fact that these aggregates are large, they're still small enough to travel from one cell to the other via transsynaptic connectivity. So here you see the receiving cell starting to express the mutant huntingtin protein aggregate. This is very toxic, and it creates degeneration, and it leaves behind these aggregates in the extracellular matrix of the graft. Here I'm talking, of course, in the context of the graft, but this could be true just in the brain of Huntington. You could have a transsynaptic propagation of the mutant protein. And sure enough, 1 month after we published the data, there was a very, very elegant paper published in Nature Neuroscience where they showed exactly this, using 2 models in vitro and 1 model in vivo, so in brain slices of animals, that the mutant protein could be transported from one neuron to the other. But if this non-cell autonomous mechanism, that is, you don't need the gene for one cell to be infected with the mutation, if this mechanism only occurred via this transsynaptic route that I just explained to you, then we shouldn't find the mutant protein in the non-P zones of the graft. Remember, these areas that don't receive cortical projections. So we think that there are also other routes of propagation besides the transsynaptic route. One of these routes is the immune system. A number of years ago, and this was actually, we just confirmed the data of other people, we took the blood of Huntington's disease patients and we looked and isolated all the cells in the blood. And we found that certain cell types, for example here the monocyte, expressed the mutant protein. Now this has important implications because in the graft and in the brain of Huntington patients, we found that the mutant huntingtin protein was also expressed in perivascular macrophages. Perivascular macrophages are monocytes that have transmigrated into the brain and taken on this new phenotype or this new identity. So this is very, very important because it means that immune cells can carry the mutant protein and probably be vehicle to transport the mutant protein. So we became especially interested in this avenue because if it's true that the immune system is involved in the disorder and becomes a vehicle for the propagation of the disease, then we need to target it with specific therapies. So one of the things that we did is that we looked at the expression of the mutant huntingtin protein in all aspects of the blood vessel, in all cell types associated with the blood vessel. What you see here is, again, our little aggregates stained in green, and you can see that they're found in the larger blood vessels in the brain of a Huntington patient. We also saw the aggregates in endothelial cells of the brain vessels. We saw them in smooth muscles of the blood vessels, and we saw them again in the perivascular macrophages. In other words, the inclusions and the mutant huntingtin protein was found in every cell type of the blood vessel. We confirmed this using a mouse model, which is called the R62. You're probably familiar with this mouse. It's one of the first mouse that was created to express a Huntington phenotype. It has a very long CAG repeat, just like in the disorder, so it's a very aggressive progression of the phenotype. And in this mouse, we were able to confirm exactly the same results. That is, the mutant huntingtin protein was found in every cell type of the blood vessel. Now, having observed this, we thought, obviously, the expression of the mutant protein in the blood vessel is probably impacting the function of the blood vessel. So we were very fortunate. A number of patients volunteered to undergo a scan, a brain imaging evaluation. And what we found is that in the Huntington brain, the blood vessel and the vasculature was different from the control brains. And in fact, the arterial cerebral blood volume, so the entire blood volume, was much greater in the gray area, so in the cortex of these patients in comparison to the control. This we confirmed post-mortem using brains of people who had donated their brains, and you can see by the measure here or the figures, or the images, sorry, that there are much more blood vessels, or there are more numerous in the Huntington brain than in the control. We also confirmed this in the mouse again. You see very clearly from this image that there are more blood vessels in the Huntington mouse than in the wild type. And we further confirmed this using a technique that's called transmission electron microscopy, where you can clearly see that the blood vessels don't have the same morphology at all as the control mice. They have very small branching, as if these are aborted. They just have a different morphology altogether. So then we also thought this must also have an impact on the blood-brain barrier. The blood-brain barrier is so important in preventing toxins and other peripheral abnormal agents to enter the brain. So again, the same patients and others generously volunteered to undergo another type of scans where we injected them with gadolinium and looked at the leakage that we could find in their brain. And what we found, it was very interesting, is that there was some positive correlation between the leakage in the structures that are targeted in the disease. So for example, the striatum, the caudate, the putamen, that correlated with the burden of disease. In other words, the more you progressed within the disease, the more permeability you had in your brain, so the more chances you had of peripheral toxins entering the brain. This we confirmed also post-mortem. This may be a bit tricky to explain because I'm really getting into details of the research, but I think it's very important to go through this data nonetheless. We looked at two proteins, occludin and clodin 5. These are proteins that are involved in tight junctions. Tight junctions are very important in this permeability of the blood-brain barrier, making sure that there's no leakage. And what we found is that these proteins did not behave normally. They were downregulated in Huntington. And in fact, when we looked at proteins like fibrin, which is a very large protein, it doesn't normally cross the blood-brain barrier because it's too large. Well, it was very, very much present in the brain of Huntington's disease patients, meaning that there was some leakage from the periphery to the brain. Again, we confirmed this in the mouse. In the mouse, we were able to take these analyses a little bit further, and we saw that the tight junctions, which again are responsible for this permeability, were actually disorganized. They were shorter, they didn't have the right angle, so they were allowing the penetration, if you like, of toxins from the periphery to the brain. And surely enough, we were able to see this, all of the white cells here around the vessels are peripheral cells, and this is in the brain of an R6-2 mouse, so we were able to see more infiltration of peripheral cells within the blood vessels of the brain of a mouse with Huntington's disease. And finally, I think this is one of the most important results, is that within the blood vessels, we have these little vesicles. They're very small, but they can transport all kinds of proteins and all kinds of garbage in and out of the blood vessels, so from the blood vessel to the brain or the brain to the blood vessels and vice versa. And what we found is that there were more vesicles in the Huntington mice than there were in the wild-type mice, or the control mice. And most interestingly is that these vesicles express the mutant huntingtin protein, so they are capable of carrying this protein, this mutated protein, in and out of the blood vessels. We don't know which direction, and we don't know how this works yet, but this is a very important breakthrough in knowing that this protein can be transported from different elements of the body. So the second hypothesis that we have, and that we're very, very actively pursuing, is that we think that blood-borne cells and peripheral cells, they all express the mutant huntingtin protein, all of them. And some of these cells, because we have a leakage of the blood-brain barrier, they can transmigrate, which they don't normally do. They can transmigrate into the brain, and they're probably receiving a signal. In this case, for example, from the graft, they're receiving a signal that something is not well and the cells are degenerating. So they're called to the site of the graft, and once they degenerate, they leave behind these aggregates. And these aggregates are then free to be picked up by other cells and be infecting other cell populations. And lastly, I'm going to finish with this little set of data. We're also interested not only in peripheral cells and blood-borne cells, but we know that these cells, under stressed conditions, under inflammatory conditions, they are capable of releasing these little microparticles that we call microvesicles. This is an example of a microvesicle. Now, a microvesicle is sort of like an exosome. You probably heard of an exosome that's detaching itself from the cell, and it's big enough to carry large proteins like the mutant protein. And in fact, so far, using again the blood of Huntington patients that generously donated their blood, we were able to identify that these microparticles exist. They're in greater numbers, and not only are they in greater numbers, but they express the mutant huntingtin protein. So the last hypothesis that we have for the propagation of the protein is that peripheral cells, blood-borne cells, are able to, under stress or under inflammatory condition, release these vesicles. They bud out of the membrane, and these vesicles carry proteins like the mutant protein, and then you don't even need leakage of the blood-brain barrier. They're so small that they can go right through the blood and seed pathology, if you'd like, or seed the mutant huntingtin protein in the brain or in the extracellular matrix. So I'm going to end with this. I have two sets of conclusion. The first set of conclusion is that I want to go over the data that we collected in the patients who received a cell transplantation therapy. I want to emphasize, and I don't know if I've convinced you of this, but I think that the Huntington's disease brain is not an hospitable milieu for the grafted tissue, at least not at the stage of disease when these patients were grafted. I did not mention the details of the clinical trial, but these patients were mid-stage to late stage, and I think it's just too late. It's too aggressive at that point for grafted neurons to survive this very inhospitable milieu. I also think that the marginal and the short-lived clinical benefits observed in these patients, combined with all the data that I showed you today, forces us to reevaluate this approach in Huntington's disease, unfortunately. I'm not saying this is the case for all disorders, but I think we have to be very careful. And before people volunteer to have this type of experimental therapy, we need to sit down and review the data. We need to think about who are the patients that we need to recruit for this. Is it the early stage patients? Who is more suitable for this type of therapy? We need to review the methodology. Maybe it's just the fact that we transplanted whole pieces of tissue. Maybe single cells will do better long term. But in any case, I want to issue a strong warning in the sense that I think we do need to reevaluate all of this data and before we undertake any more experimental therapies using cell replacement therapies, and especially stem cells, where we don't know how they will behave in the brain. My second set of conclusion is that I know it's casting a bit of a shadow over this therapy, and maybe you had some hope with this, and I'm bringing a lot of caution to it, and I'm signaling a lot of obstacles that we have to overcome. But I want to finish with a very, very positive message, and the positive message is that these grafts and these brains have been invaluable in understanding Huntington's disease pathology. Since we have studied these brains, we have made such progress in understanding how the pathology occurs and how it's spread. And now we know that there are non-cell autonomous mechanisms underlying the propagation of the mutant protein. So this is very important. It's not just the gene that drives the pathology. So despite the fact that we don't know to what extent these mechanisms contribute to disease onset and evolution, it certainly opens up to a whole new way of looking at this disorder and identifying new targets to develop therapeutic strategies. I'm sorry, this slide shouldn't have been there. So now, of course, I want to tell you how committed we are to these projects. I've been working in the field since I was a graduate student more than 20 years ago, and I see how much progress we've made in the field of Huntington's disease, and I want to continue these efforts. So now we're doing a lot of work using animal models, but also continuing to interact with the patients, getting blood, and trying to study these phenomena. So I have a number of collaborators. I've been able to rally many of my colleagues into this endeavor. So we're doing, for example, very neat stuff like 2-photon imaging to look at the propagation of the mutant protein. We're looking at the mechanisms of protein spread in very neat models of in vitro systems of the blood-brain barrier. We're able to model the blood-brain barrier in vitro, so this is quite spectacular. And we're continuing all this leakage work with Roger Barker at the University in Cambridge to try to better understand this and developing, of course, therapies in the long term. So I just want to end by acknowledging the work of so many fabulous people. This is my team at the moment. The people in bold are the people that have been driving this project, especially Julia Chisbani, who's also funded by the Huntington's Disease Society of America, so we're very grateful for that. She's done a huge amount of work on this project. Also, Janelle Drouin-Wallet, who's now a postdoc in Roger's lab. She's a former student in my lab, and she's done fabulous work on that as well. And all of our collaborators, which are putting tremendous effort into this and are believing in these new hypotheses and the development of therapies. So I'm going to end right here, and I'm truly looking forward to your questions and your comments. Thank you very much.
Thank you so much, Francesca. That was really, really great presentation. It's a very deep topic, and I think you really covered it very beautifully. And it's, for those that may not be familiar, I'm in awe. This was a massive amount of work that she just presented, and I just want to thank you for presenting it in a very easily understood way. So, folks, if you have any questions, please, you can type them in at any time. I'll go ahead and get started. I'm interested in and intrigued by this approach of stem cell therapy has been around for a long time. The idea, you highlighted a number of studies that were done more than a decade ago. Has there been any breakthroughs or insight as to what is the state of the art or the best path forward in terms of the type of cell, whether it's a slice or a section or a dissociated cell for these types of treating brain diseases? Has any progress been made in that area, Francesca?
Okay, so in the trials that I've shown, unfortunately, all the European groups have not been allowed to do postmortem analysis, so we've been very limited to just the clinical data. So obviously that's unfortunate. And I think that because it's such a limited number of cases, it's very, very difficult to derive anything solid in terms of what is the best methodology. Of course, we've learned from animal model work and so on, but it's difficult to translate this to the human disease, and each patient is different stage, is responding differently, is submitted to different regimens of immunosuppression, which is also very important in the survival of these grafts. So it's difficult at this point to know, but if we look at what's been done in Parkinson's disease, where they've had much better success, I mean, I've seen some cases where the improvement is night and day, and in fact, there's a huge clinical trial going on right now in Europe, led by Roger Barker, where 60 Parkinsonian patients have been recruited over 5 different centers and are now undergoing this procedure. They have opted for single-cell transplantation. They have opted for that because they seem to survive better. They have opted for younger patients because they respond better. The only problem now is that also in the Parkinson's field, we know that the mutation of the pathological proteins spread from cell to cell. So they will be facing the same problems as what I'm showing today. But their argument, and I think it's valid, is that it takes a number of years for this to occur. So we shouldn't stop ourselves necessarily from not doing this, given that it may take 5, 6, 7 years before we see the propagation of the mutation. So we do have to also keep that in mind. So I think there are possibilities to improve this technique. I think that in Huntington personally, and this is of course my opinion, I don't want to drag any other colleagues into this, but it will be difficult because the pathology in the brain is what it is. I mean, it is quite severe. So one advantage that we have, though, is that we have a genetic test. Could we imagine doing transplantation in people that have the gene but have not started to show correct symptoms, the clinical symptoms? Possibly. So I'm not completely ruling it out, but I think we have a lot of work to do before this is, offered to a large spectrum of patients.
Great. Thanks. A couple questions. They seem to be from the audience, somewhat related, and touching on the idea that Huntington is being transferred via the blood vessels. Mm-hmm. So this question is that, the community is often asked about blood donation by people with HD, and considering your evidence that mutant Huntington can travel through blood and propagate elsewhere, should we be dissuading people from being blood donors?
This is such an important question. I had a colleague calling me last year. There was a a Huntington's disease patient that had offered, I think it was his liver or a kidney for a transplant to someone else, and they were concerned that the pathology could spread. And before a couple years ago, we never asked the question because we were really convinced that it was you needed the gene in order to express the pathology. And I think that everything we're seeing now, and this is not just the work of my group, in the last month there's a couple more papers that have shown that the mutant huntingtin can propagate in vivo system in live animals. So we really need to reconsider this. I think that there's a real danger and warning there that mutated cells could infect other cells.
More about the kind of the mutant huntingtin and wild type or non-expanded huntingtin protein. If the mutant protein can spread from the bad to the good cell, the uninfected cell, why can't the non-mutant, non-expanded, or good protein spread in a similar manner? If we can silence the bad protein and amplify the good protein, could the good protein spread from cell to cell? I guess they're trying to look at the other.
This is an interesting question because when I think of the problem, I think, and I have bronchitis now, so this is where the example is coming from. If you have 5 people that have a very bad cold and you put 1 healthy people in the center, you'll probably have that healthy person develop the cold as well and not the other way around. And I think in the scheme of the graft that I showed today, the healthy cells, the ones that we transplant, they're not in a huge number. I mean, we're not replacing the entire brain. We're putting, you know, infinite numbers of these cells, like very small numbers of these cells in a massive environment where everything is contaminated. So it's difficult to imagine that this small amount of cells would themselves be able to contaminate in a good way or reestablish the health of all of the other cells. I think the other scenario is more likely because of the outnumber and so on.
What about, you know, we're very focused on, and the community is very well aware of gene lowering approaches for Huntington's disease, and one of the big issues with this, whether it's, you know, the ASO therapy that ISIS is working on with Roche or other companies planning, it all involves delivery of gene silencing reagents or drugs directly to the brain. Would your observations maybe make it feasible to deliver these gene silencing or agents peripherally in the blood?
I'm personally very excited by these therapies, to be honest, and not so excited by, of course, this targeting of the brain. I know that the ASO trial will be targeting somewhat the peripheral system because they'll go into the spinal cord. But I did have this discussion with someone before, and it would be, in my opinion, a good idea to start targeting the periphery. But they were telling me that it was much harder to do this because it wouldn't be as specific as going exactly where they have this massive neuronal loss. So I think eventually, and they have also this problem of crossing the blood-brain barrier, but if we are showing indeed that there's a leakage of the blood-brain barrier, then it could be used in the periphery. Now this blood-brain barrier leakage is somewhat subtle, and it seems to correlate with disease progression. So in early stages you don't have the same type of leakage that you do later on, so you wouldn't be able to use this as a tool to better transport your peripheral therapy into the brain. So I can understand why they're going straight into the brain, because they have this problem with the transport through the blood-brain barrier. But I think these are exciting times, that this is an exciting way to tackle this problem, and that eventually we do need to think of applying this to the periphery, and very early on, in fact.
Great. Just a quick question, and we have maybe time for just a couple more. I want to be cognizant of everyone's time, but can you just comment on, I think there's a little, they wanted clarity about how the stem cell replacement compare the stem cell replacement data to gene replacement. How is that necessarily different?
Okay, so I just want to make sure everybody understood that what I talked about today is fetal tissue. That means that when you take the dissection of the tissue in the fetus, these cells are already committed to an identity. They're either dopaminergic, they're GABAergic. The only thing they don't have is that they haven't made the proper connections yet. So we dissect them, they're dopaminergic, the GABAergics, we put them in the brain and we hope they make connections. With stem cells, we're at a much earlier time point of development of these cells. And that's where I think that we need to be very careful, because a stem cell is a cell that's still dividing and we don't know how it's going to behave in the brain. So if it starts dividing in an uncontrollable way, this will create tumors. And then we're way worse than we were without the transplant itself. So we have to be extremely careful with that. Now with the gene therapy, other people are trying to inject cells that will release trophic support, for example, in a natural way. And I think this is it's a clever idea in the sense that in the brains of these patients, you lack trophic support. We've seen these similar trials in Parkinson's disease where they've injected neurotrophic factors. So having a vehicle like a cell delivering this naturally is worth pursuing.
But the same thing might be true, right, with those cells delivering a drug or a trophic factor. They could be infected by the mutant huntingtin.
Yes,
you would expect to be infected by the mutant huntingtin.
Yes, at any point I think they will. But again, the data I showed was 10 years post-transplantation. We never saw this in the patient that passed away 2 years after the transplant. So there is a time frame that you need for this propagation to occur. So does it mean that we would transplant periodically to replace the cells that have now been infected? Would that be a possibility? Perhaps. But ultimately, I think any cell type would be infected, unfortunately.
Well, thank you, Francesca. We ran a little bit over, and I apologize if there are a few questions that we did not get to answer, but please do, I'll try to get back to you, answers either from myself or Francesca after the webinar. I apologize for that. But thank you again, Francesca, for taking the time and presenting this really important topic to the community. I appreciate it.
Thank you so much, and I'll be happy to take questions via email if this is necessary.
Okay.
Thanks
Thank
again.
you so much. We'll talk to you soon.
Thank you.
Bye-bye.